I really want to do today is come in somewhere between a boring genome, yet another genome, and talk to you about potentially actionable genomes to see what we might be able to do next. And I'd first like to acknowledge that this is a team effort. I'm just the lucky one up here who gets to present it to you. So we have, especially I'd like to acknowledge Dennis Dean and Bob Coopersmith, who are also project managers on this. We work very closely with Saiju and his team at the VA, and also Dennis, Brandy, and Devin for making this possible. We also have the engineers who are building the pipelines and building the common workflow language, allowing us to make this data actionable. Sorry, this mic's just... So I want to start with a little bit about precision medicine. I think most of the people in this room know about precision medicine. For the few people who might not, this is a picture of President Obama announcing the Precision Medicine Initiative. And to give a concrete example of precision medicine, let's imagine I go to my oncologist and she says, Jack, you have leukemia. What we're going to do is give you chemo A because 60% of people respond to that chemotherapy. Now, as an alternative, I could go to my oncologist and she might say, Jack, you still have leukemia, but we've noticed you have this collection of variants. And there's another group of people who have the same variants. They also had leukemia, and they responded very well to chemotherapy B. We're going to give you chemotherapy B instead. That's the promise of precision medicine. That's delivering the right therapy specifically tailored for me. Now, I'm pulling these numbers from the air. This is very much a numbers game. There need to be enough people like me with similar variants who also had leukemia, who also tried these different chemotherapies to be able to make that kind of a statement. Fortunately, we're in the era of huge genomics projects. So here we have a log scale. At the bottom is 1,000 genomes, which was a huge effort. But now, a whole order of magnitude higher is TCGA. Jumping up another order of magnitude, we have Genomics England and Genome Asia. There'll be 100,000 patients in those databases. Qatar will break 300,000, and the Million Veteran Program is up at 1 million subjects in the database. Just a few weeks ago, while I was preparing this talk, AstraZeneca made a blockbuster announcement. They're going to hit 2 million patients. So what does that mean for medicine? Well, it could be really exciting times. At Seven Bridges, we're involved with the TCGA in our cancer genomics pilot, in Genomics England for graft genome technologies. And what I'm going to focus on today is the Million Veteran Program, or MVP. First, I want to give a disclaimer. This is a collaborative research and development agreement with the VA. MVP is a much larger project at the VA, and we're collaborating on a small part of it to develop technologies. I'm going to cover 3 things in this talk, a little bit of background about MVP, and then 2 of the things that we're doing within the CRADA to empower analysis and develop new tools for research. So the Million Veteran Program, the goal of this program is to partner with veterans to understand how genes affect health. And this is coming right from their webpage. A couple really interesting things that pop out of this project. First, it's a single provider health care system that everybody is coming from. And this could do nice things for harmonizing annotations and phenotypes. Second, veterans are volunteering to help other veterans. And by providing their samples, there's going to be more of a percentage of the diseases that veterans are suffering from, really creating a nice database for them. The current numbers are as follows. There's about 400,000 genotyped arrays. These are AFI chips with more than 700,000 variants on them. There's 30,000 whole exomes collected and 2,000 whole genomes. There are currently 8 scientific groups within the VA that are investigating different diseases. So there's one looking at schizophrenia. There's one looking at PTSD. There's one looking at hypertension within the data that they have today. But by 2020, they expect to hit 1 million patients. And importantly, there'll be a relatively higher percentage of whole exomes and whole genomes in that database. What does this mean to have so many patients? What's a concrete example of why this might be useful? Well, traditionally, African Americans represent a small fraction of participants in a study. They're underrepresented. The Jackson Heart Study from Jackson, Mississippi, was looking specifically at heart disease in African Americans. Still, they only reached 5,300 patients. With the current numbers in this paper I'm going to talk about in the next slide, There's over 30,000 African Americans in the MVP database right now. And if these stats hold and they stay on target, by 2020, there'll be more than 130,000, providing a real chance to do precision medicine in that community. Further, coming to the boring genomes, coming back to that, most of the MVP cohort is over the age of 60. So these are patients that have their genome sequenced. They're also probably, more likely at least, to be suffering with hypertension, with arthritis. They're presenting diseases now to build cohorts to look into. If precision medicine, and this is an if, this is not saying that it will, but if they're able to develop therapies in this group, this has a real potential to reduce healthcare burden. There's a really nice overview. If you go into Google Scholar and just Google Million Veteran Program, it'll come up first or second by John Gaziano. If you happen to be in Boston at Biodata World, he's going to give a talk on this topic. But it gets into all the details you might have, all the questions you might have about the data. And the data is great. It's a prerequisite. We absolutely need it to move forward. But there's more than that. We need to enable you to use the data, or researchers to use the data. And there's sort of 4 key challenges we have to address within the CRADA. First and foremost is security. Patients' personally identifiable information, personal health information must be protected and kept confidential. Once it's secure, let's say I want to give you a data set. I want you to analyze it. How would I do that? This is not passing around flash drives and things. This is 200 gigabyte files. So I don't know how I get it to you. But even if I did, I don't want you to melt your laptop trying to compute on it. So you need the resources to do compute. And let's follow the thought experiment farther. So I've given you the data, I've given you the data, the guy in the back checking email has the data, everybody has a copy, everybody's doing analysis. And we try to harmonize the results, but you all use different processing. This could introduce artifacts which would be very problematic to be able to understand anything from the results. So the next 2 topics, we're going to talk about how we're empowering analysis, basically while respecting the data security, meeting those last 3 needs. Conceptually, how we do this is we bring the compute to wherever the data is. And this happens through 2 mechanisms. The first is Common Workflow Language, or CWL. This is a community-driven, open-source language that allows portable, reproducible, and scalable analysis. How many of you are familiar with CWL? Quick show of hands. Nice, we got some people, that's great. For those of you that aren't, you could imagine it as a JSON or a text file that basically says, I want to do this analysis, these are going to be my input files, these are my configurations, and this is the code I want to do it. So if I have that file or you have that file, you know exactly what I did. And for reproducibility, you could imagine, you get an edit, a revision to a journal article back. They want you to redo analysis you did in 2013. If you had this, it would be push button. Go ahead and do it, which would be awesome. But it's not just CWL. We pair CWL with this whale. So what is this whale about? Well, this represents a Docker repository. And this is important because if you remember in the CWL, I said there's a description of what I want to do and the code I use to do it. If we keep the code up in the repository, I can easily share it with you. I've given you the text file that tells you what to do. It's also pointing to a particular piece of code in that repository. So I can run it, you can run it, you can run it with no problems. We're going to exactly reproduce the same analysis. The final piece of the puzzle, putting these together, is something, a suite of developer tools we call Rabix. And importantly in here, there's an executor. So now I've given you a description of what I want to do with CWL. I've given you the code in a Docker file, and this executor is going to say, OK, take that and run it on my laptop, run it on my desktop, run it on a cluster. But it's going to be the same exact code in every single place. And if you want some more information, and you happen to be going to ISMB in 2 weeks, Gaurav is giving a really nice workshop on this, digging into the CWL and Rabix. To give a concrete example for the VA, let's say we have everything in red here is the security of the VA. The data is never leaving the VA. That way we know it's remaining secure. But we have a couple different VA research sites. So we have location A and location B. and we have a researcher in location B. She wants to do something. She knows that the data is over there in location A. With these combination of technologies, she can write up a CWL description of, here's the analysis I want to do. She sends it to center A, but importantly, this size is like kilobytes, maybe megabytes of data going over. You don't have to transfer those 200 gigabyte files. They pull down exactly the code, do the analysis, and she gets the results. And it wouldn't matter if she would have done that in location B, if the data was there, if she would have done that in location Z. It's always going to be the same at every place. As a concrete example of what we're trying to do there, of what we're benchmarking this with, it's a gold standard whole genome sequencing pipeline. That's this terrifying sequence of dots here, terrifyingly complex. Each one of the blue dots is one particular tool, so there could be an aligner, a variant caller, a QC, but that is all described in CWL. And what it's going to do is harmonize the data. So if we run this on 100 different genomes at different locations, we would expect to have, we will have exactly the same processing. And from a logistics standpoint, it's nice because the output is VCF files, which if we needed to transfer data, small and easy to do. So that's enabling compute. The next thing I want to talk about in the last few minutes is graph genome tools. How many people are familiar with graph genome in the audience? Another big percentage. This is great. So at 7 Bridges, we really love graph genome, so much so it's in the name of our company. And for those of you not familiar, I can give you a quick run-through of a graph. So here is a reference for a population, and we're just looking at 6 base pairs here. Nice short sequence so we can put it on the slide and understand. And what this represents is what happened most frequently. When you get into a graph, you start to annotate the percentage of times that you saw this. So we saw this particular sequence 60% of the time. There was a SNP rate there 10% of the time. And another 30% of the time, we didn't follow either one of these pathways, but there was just a deletion, and we jumped over this whole thing. So now we've captured the variance within the population instead of collapsing it to the mean. It's a really nice representation. But think about some of the possibilities from this. If we have a whole population of people in there, we can capture the variance, but as long as we remove the paths, we've anonymized the data. So now you can capture all of the variance without jeopardizing patient privacy. Second, think about it for compression. As we get up to 1 million, 100 million people in there, we're only storing one reference basically and any nodes coming off of it. So this can incredibly compress data. For more information, if you're in a national project, I'd encourage you to Google, it's probably too small on the screen, Google Graph Genome Day. We're releasing our graph tools at the end of September, and there's a very cool release in London. I'm just going to finish off with 2 practical examples. Graph is a nice structure, but what can it do in this space? What might it be able to do? So I've made here 3 toy population graphs. I have a European graph, an African American graph, and a Hispanic graph. You can see they're different colors. They have different structure. You would expect this from different haplotypes. So this is a nice sanity check. We see different structure in different populations. But let's bring it to a concrete example. What if we knew that a certain heart medication worked really well for Europeans and worked really poorly for African Americans and Hispanics? You could start to think about graph operations, things you might do with the graph. So let's combine these 2 populations where the heart medication doesn't work, and let's sort of do a differential and try to understand why doesn't it work in one and it works in the other one. And what this might do is highlight differences in the graph structure where precision medicine researchers can take these tools and search for actionable variants in that space. As a second example, as I brought into the introduction, the graph can represent the percentage of variance, but why don't we overlay this with the phenotypes? So let's say, to start, you come through CGTCCC. That's going to be associated with a certain distribution of phenotypes. Here we're going to call green healthy, blue is hypertension, and yellow is COPD. Well, now if we take a different path, we take the G here, we can overlay the phenotype again, and right away you see, oh, it's different. We have slightly less healthy people in green, the same number of people with hypertension, but all these new phenotypes jump in. So that might jump out at you and say, oh, an association study would be great there. And maybe it would be. Maybe this tool would enable that. Further, think about search. Now you have all of the genetic structure of the population and also all of the phenotype distribution of the population saved in this graph. This may open new possibilities for researchers. So I've talked about the Million Veteran Program, a quick overview of what they're doing, and two of the research and development topics that we're working with the VA on in our CRADA. We really are excited about the MVP. We believe it has an opportunity to be transformative for precision medicine, and we're actively collaborating with the VA to keep the data safe, enable reproducible workflows, develop some gold standard processing techniques, and create graph tools for researchers to advance precision medicine. I really thank you all for your time and your attention. I'm happy to take a few questions. In case we run out of time, I'll be at the booth 426 later today.